Hey folks, and welcome to another edition of Zog Science. Today we're going to be finishing up development by talking about plants. So just a quick overview. Development for plants occurs throughout the life cycle. It doesn't ever kind of stop like it does in animals when they reach the adult stage. And what powers this growth is the meristem. And there are a couple different types of meristems, but basically it is a permanently undifferentiated embryonic tissue, and it is found throughout the plant, and it is the source of all plant growth. So you've got the apical meristems, which are at the tops of the plant. You've got axillary bud meristems, which are going to be creating buds, and then you've got lateral meristems, which are the vascular and the cork cambium that are actually inside the plant, and then you've got your root apical meristems. So a quick note about primary versus secondary growth. Plants grow continuously, but there are different types of the growth. The first is the primary. This is the vertical growth, which is driven by the apical meristems of the roots and the shoots. So that's going to be the stems, the tops. Whereas the secondary growth is horizontal growth and is driven by the lateral meristem found in vascular and cork cambium. So what happens first is the primary growth elongates the plant, and then the secondary growth comes along and expands the plant. So what happens at the shoot meristem, which is the very top of the plant, you've got a spot of all embryonic tissue that is just dividing, growing, creating new cells, and as it does so, it is building the plant up. It is also going to be producing leaf primordia, which are young leaf kind of precursors that will eventually become leaves. And again, it's just kind of building those up. Building those up as it goes along. You also see the axillary bud meristems, and that will produce lateral stems during the secondary growth process. Now, those only will function once the meristem, the apical meristem, gets far enough away, which makes sense, right? You want to be elongating the plant first, getting it nice and tall before you actually kind of force more lateral movement, or not lateral movement, lateral growth to occur. Then down in the roots, you have another meristem, which is going to be elongating the roots, putting them deeper and deeper into the ground. This is whenever you've looked at onion cells for mitosis, whether that was in freshman biology or in AP biology. This is what you were looking at, was a meristem, and that is the kind of the zone where the most cell division is occurring, and that's what is actually elongating the root. And then you've got a root cap that is kind of protecting that zone, and because it's pushing through the ground, right? So you want to make sure that it is not being messed with. Once the primary growth finishes, so either from the shoot or from the root, the secondary growth is going to occur, and it's going to occur because of the cork cambium and because of the vascular cambium. So the vascular cambium is what actually produces the layers of the xylem and the phloem, and the cambium produces the xylem first. So it produces xylem, and then as it does so, it is growing the tree out laterally, and then it produces a phloem, and it produces xylem, produces a phloem, produces xylem, produces phloem, so on and so forth. So you actually will always have a little more xylem in general than you will have phloem. Now, what's kind of interesting with this is that as the wood kind of goes along, it is getting larger and larger, right? It's producing the xylem and it's producing the phloem, and it is elongating all of these sections kind of at the same time. So the cork cambium is what produces the protective bark. So the cork cambium is going to be right along the outside of the tree. Now, the bark does consist of the phloem, and it is kind of right around the outside of the vascular cambium itself. And then the xylem is going to be towards the inside. So what happens is that we have, in the beginning of the growing season, so starting in the spring into early summer, you have what's known as the early wood, and at that point, the xylem that are added on are much larger, and that's why you can kind of see here these much larger kind of holes. These are the xylem that are much bigger. And then later wood is much smaller. That's towards the late end of the season. Growth is a little bit slowed. And then that growth occurs in a yearly cycle. So then here would be our vascular cambium that had been producing that growth, and it's also producing the phloem on the other side over here. and then again the cork cambium producing the cork that is along the very outside of the tree, along the outside of the bark. So wood production, again, wood is produced by secondary growth, and wood is incredibly important, right? We use it in so many things that we as humans have done in terms of building. Um, so again, the vascular tissue near to the interior of the tree becomes non-functional. So after probably after 2 years of it being functional, it dies and it becomes non-functional, and we call that the heartwood. Whereas the vascular tissue towards the exterior is still being used, and that is called the sapwood, and that is where the actual water will be flowing up through the xylem. The heartwood would actually rot pretty easily if air was able to get into it, but fortunately trees are they're not going to allow that to happen. And then you've got your vascular cambium that's producing the xylem on the inside, the phloem on the outside, and then you've got the periderm on the outside being produced by the cork cambium. Now we can use these tree rings, these periods of growth, in order to study time. And just kind of as a side note, pine trees, wider rings equals hotter years. You can kind of imagine in general that having a wider ring would indicate, good growing conditions, right? Because the tree was able to add on a lot of things. Whereas a smaller ring would be, poor growing conditions, whether that was, some sort of drought or maybe there was some sort of parasite that was eating the leaves. So there's, a lot of different factors that could be affecting it, having it be lower growth, but that's, the trend that we look at. We also know that every single ring is going to have, a light and a dark layer. The reason being that the light layer is the early growth, the dark layer is the late growth. Now, as plants are developing, the symmetry of the cell division is incredibly important. You can either have just the cell division occur symmetrically, where all of the cells share the same fate, or you can have asymmetrical cell division, which is how we actually get the production of the guard cells that occur around the stomata. It's less important in terms of overall plant structure. So you could have asymmetrical cell division that can occur in like the leaves, and it can create a leaf that looks fine, even though the actual cells aren't like symmetrical, that nice kind of boxy shape that we tend to associate with plant cells. Now, why this is important is that in early development, almost all of the cell division is asymmetrical because you're producing all these different types of cells that need to perform different functions in order for the plant to actually survive. If all of the cell divisions were symmetrical, the plant would not develop properly. So if all the cells continue to be this kind of embryonic tissue that was able to perform all functions, well, you would never actually get a plant developing properly. So asymmetrical cell division, where the cells that are produced by the plant having different functions, different fates, is actually incredibly important for the function of the plant and its growth. We can also have issues arise because of manipulation of the genetics of the plant development. So for example, this is a tomato plant. These are the normal plants over here. If you overexpress one of these genes, which is called the knotted 1 gene, it can lead to these kind of super compound leaves where you get all these little tiny leaflets. It's kind of interesting that you can have such a radical difference in the leaf structure just from the overexpression of one of these genes. But it's very similar to what can happen with animals and what can happen with humans, right? Genes play such an important role in the development of all species. Just like if a homeobox gene was messed up in an animal, it wouldn't be able to develop properly. Similar things happen with plants. Plants also go through the process of differentiation. The little bit of difference is that animal cells tend to differentiate based on what is known as lineage mechanisms. So basically, you have inherited as an animal cell a particular set of chemical precursors or chemical signals that are helping to determine what type of cell you're going to become, whereas a plant cell is differentiated based on its position. So a cell is not necessarily destined to become something until it gets into a particular position. So for example, the cells that are blue here, they're expressing a particular gene, and those are going to develop into a root hair because of their location, whereas the other cells around the outside, they're not going to develop the root hair simply because of their location. It's not that they were predestined to before they actually got into that position. So finally, genetic control of flowering. You can see that here we've got the sepals, the petals, the carpel, and the stamen that have all formed normally. However, you can have abnormal development that can occur with mutations, mixing of the genes, all that kind of stuff. You can get various combinations. In this case, we only have petals and sepals, but you could also get only carpels and petals, or you could only get steeples and petals. Like you can get all kinds of different combinations. Just depends on whether or not all of the genes that are necessarily kind of get passed along. So very similar things to what we've seen with humans and other animals. You can also see that stuff in plants. So thanks for watching. Next time, I believe we're going to be talking about animal responses to their environment. So thanks for watching, and I'll see you next time.